Hey guys, welcome to GeeklyEDU. This is me, Sona, saying, and today we are going to talk about genotype and phenotype, two terms which often people get confused about. So, what is the genotype? Genotype is nothing but the set of alleles, set of gene variants that you possibly have. Okay, uh, it could be same. You could have the same alleles, or you could have the different alleles. Why? Because we are diploid. We have two copies. We receive one copy from mom. Let us say these are 3 chromosomes. Let us say we are an organism with 6 chromosomes, just assuming we have to have 3 from mom and 3 from dad. We, although are 2 into 23, we have 23 from mom, 23 from dad. And just for this example, we are going to take 3. So the yellow ones are homologs because they have genes that affect the same characters. So let's say, for example, the yellow one has the gene for blue eyes. At the same location, you'll have a gene that affects the blue eyes. It might be that you have got the same recipe, like blue eyes from both mom and dad, or for dad, you might have a black eye. So if you see the sequence, this is just a random example. A slight change in just one nucleotide might make all the difference that you need. Just one nucleotide can affect the change, or it might just not as well. So, what are these two things? These are two different alleles. If you need more information about it, you can check the gene and genomes lecture as well. Now, so when you write those alleles, you're trying to represent the genotype. So A blood group has a genotype IA, or IA small I. Why there are two possible genotypes? Because the external visible form, the measurable form, we use the word measurable, not visible, because sometimes the blood is same. The blood, A blood and B blood doesn't look different, but they are biochemically different, right? So they are the same genotype and they are the same phenotype. The external manifestation is also the same, right? But look at the B blood group. It could be IB. It's a different genotype, right? So the first two were the same genotype, same phenotype. Sorry, different genotype, same phenotype. And the second two are different genotype, different phenotype, right? Now, why we use measurable? I hope you remember, because we measure this biochemically. Okay. Now, how do we define phenotype? Just to summarize, it is the manifestation, manifestation or the measurable form, of the genes that you possess. Now, genotype exerts the limit on phenotype. What does this mean? Let's say P is big T, that means it has the alleles for tallness. That doesn't mean it will become tall if you don't water it, right? If you don't water it, it won't become tall just because it has the genes. So genes set the limit. If it's tall, it is supposed to be tall, but only if you water it. Okay. So genotype must be understood clearly. It is the limit on the phenotype and not the compulsory manifestation. Okay. Now, when we talk about phenotypes, we must also be careful. Let's take a case of sickle cell anemia. What is sickle cell anemia? Your hemoglobin is there inside RBCs. Now, hemoglobin is a protein which has globin chains in hemoglobin, right? Remember the central dogma? If not, check out the genes and genomes video to understand that. Okay, so gene goes to mRNA, goes to polypeptide. Now, if a single AT sequence changes to 2A sequence, the polypeptide has a different amino acid. Glutamate changes to valine, and because of that, the hemoglobin can clump down, and it can lead to formation of sickle cell RBC. Okay, and this happens only when the oxygen concentration does go down, and adults cannot survive this. Now let us take HbW, means the wild type Hb, and HbS, the normal hemoglobin. Let us see what happens. HbW, HbS, 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 and HbW, HbW, 3 different combinations. So let's first imagine normal Hb, 2 different alleles, They both code for normal hemoglobin, and you have a normal shape RBC. It's around just like a donut. What if I have one wild allele and one sickle cell allele? What will happen then? Well, half of the hemoglobin would be normal and half would be abnormal. What does that mean? That means this is codominant. So, dominance and recessiveness should be used for phenotypes, not for genes. Because what we can say is when you have a mix, the person survives. But when we have both abnormal copies, you'll have both abnormal hemoglobins and the person dies. So, if you see at the molecular level, it is codominant, means both the alleles are expressing themselves. So, phenotypes are always are linked as dominant or recessive. But remember, it is the phenotype that should be addressed as dominant or recessive, not the genes, not the alleles, right? So, if you see at cellular level, it's incomplete dominant because the RBC would not be too much sickled because half of the hemoglobin is normal. But if you see at organismal level, it is recessive. So characterization of phenotype is very, very important. It depends on the perspective and the level that you're examining it at. So let us zoom out a little bit and try to understand all about what we read. Okay, we have read about genotypes, we read about alleles, and we understood genotype and phenotype. We understood how are they expressed, right? We understood that gene exerts limits on phenotypes, and we also understood that uh, phenotypes should be addressed as dominant, recessive, incomplete dominance, and not the genes. I hope this helps.
We hope this video was helpful. To see more biology topics covered, click on our channel and don't forget to subscribe.